We'll be using 2 lectures to cover Chapter 15, so today we're going to talk only about pages 493 to 509. The name of this chapter is Intracellular Compartments and Transport, and we're really going to break it along that line. We'll talk about intracellular compartments first in this lecture, and then we'll save intracellular transport for the next lecture. So more specifically, today we'll be going over the organelles again, just as we did before, but just in a little bit more detail. And we'll focus most of our time on the endoplasmic reticulum and talk about everything that goes on at the endoplasmic reticulum and everything that comes from it. We'll then talk about how proteins are sorted at the Golgi complex, not in any great detail, but at least we'll refer to it. And we'll circle back to signal sequences on proteins and spend quite a bit of time talking about how these address labels, these address sequences, target proteins to their specific locations in the cell. And that will lead us directly into where these proteins go once they're targeted, and more importantly, how they get into each of the individual membrane-bound organelles. And we're actually only going to do a few specific organelles today, and you'll see why as we get there in the lecture. So, when we're talking about a single cell, a living cell, a healthy cell, every single part or aspect of that cell contributes to its survival. A cell is really nothing more than this hugely complex, complicated orchestra, this choreographed set of activities and functions that all work together to keep the cell alive. Another way to say that is all of these parts of the cell work together to hold entropy, or to hold chaos, to hold the universe's tendency towards chaos at bay. But we shouldn't mistake that coordinated orchestra to mean that everything is happening the same way at the same time in the same direction. Just because every part of a cell contributes to a reduction of entropy and contributes to life, that doesn't necessarily mean that every part of the cell is coordinated with every other and doing the same thing at the same time. Every living cell has an ability to make glucose through a process called gluconeogenesis, while other pathways, enzymatic pathways in a cell, can break glucose down for energy, starting with glycolysis. We've seen in this class how some messenger RNAs are degraded, while other messenger RNAs are being transcribed. Molecules are being released by a cell constantly, but the cell is also bringing molecules in from its outside environment. All of these are examples of opposing pathways, things a cell can do one way or the other. If the cell wasn't made up of individual compartments, if the cell didn't have compartmentalization, cells would spend a lot of time doing things and then simply undoing them again. Cells would spend a lot of time making glucose only to break that glucose down, making RNAs only to break those RNAs down, releasing molecules only to bring them back in. And this would be a waste of energy. I kind of alluded to this and drew a crude diagram of this in class the other day, but this is what we mean. The cell would fall into this pattern of just running in place, expending a great amount of energy, getting nowhere. And obviously, that is not good for life. So, we need these compartments. We need these organelles. We need these membrane-bound regions of the cell to keep these processes apart. If we break those compartments, if we lyse a cell, if we destroy all of the membranes of a cell, and let all the compartments and all the enzymes and all the pathways of the cell intermingle, it becomes chemical chaos. For a cell to operate effectively and efficiently, we need organelles, we need membrane-bound organelles, we require compartmentalization. So we've gone through the organelles already right before the last exam, so we know in general what they do from this class and from others, but we're going to go into a little bit more detail today. The organelle membranes, those are the membranes for each independent specific organelle, they're highly specialized, and we've talked about that already. They contain a unique composition of lipids, a unique composition of proteins, and, as we just said, those membranes around organelles allow for organelles to be compartmentalized. These membranes can create enclosed compartments where enzymes can work inside that compartment in peace and in quiet and do the biochemical reactions that are specific to that organelle. And because things have been compartmentalized, even if there is some opposing pathway with opposing reactions occurring elsewhere in the cell, it doesn't interfere with and waste the time of these reactions because everything is doing what it should, where it should, in these segregated compartments. We've already said it, but all organelles in the cell are surrounded by the cytosol. The cell itself is made up of a lipid bilayer, a cell membrane that surrounds it. The nucleus is the most prominent and the most important of all these organelles, and the nucleus is surrounded by its own specific and unique membrane that we refer to as the nuclear envelope. The nuclear envelope is actually a double membrane, and that's different from a bilayer. A bilayer is a single membrane made up of two different phospholipid layers. The nuclear envelope is a true double membrane. So maybe to drive that home, there are 4 bilayers in the nuclear envelope. The nuclear envelope is completely impenetrable. You may remember from previous lectures that things like water and oxygen and carbon dioxide, ethanol, these things can readily diffuse through the cell membrane. They don't require transporters. Not true for the nuclear envelope. Nothing floats through the bilayers of the nuclear envelope. The only way to get into or out of the nucleus, the only way for the nucleus to exchange things with the cytoplasm is through these specific structures called nuclear pores, and we'll talk quite a bit more about them later on in the lecture. Nuclear pore is a very complicated structure. It's made up of many, many different protein subunits, and this is the sole entry point into or out of the nucleus. Interestingly, the outer membrane, the second membrane of the nuclear envelope, is continuous. It's connected to, it's part of, it feeds into the endoplasmic reticulum. So the nucleus and the endoplasmic reticulum are really just extensions of one another. They're not really separate structures. There's no break between them. The endoplasmic reticulum, as we've said before, is a complex collection of interconnected membrane tubes and membrane stacks where new membranes are made, as well as proteins that are destined for secretion. We've said it before, but the parts of the endoplasmic reticulum where proteins are being made is the rough ER. It appears rough because of ribosomes coating it. The ER that is devoid of ribosomes is referred to as the smooth ER, and this portion of the ER is more involved in membrane synthesis. We'll talk about the rough ER later as well, but for now, remember that those ribosomes are what make it look rough, and more importantly, more specifically, those ribosomes that stud the rough ER, they're stuck to the cytosolic side of the endoplasmic reticulum. So those ribosomes, they are on the cytoplasm side, not facing the interior of the endoplasmic reticulum. And when those ribosomes are making proteins, those proteins are being fed into the endoplasmic reticulum through the membrane. And so indeed, if we were to see the rough ER close up, this schematic represents it quite nicely. We have the endoplasmic reticulum membrane here. You can see these ribosomes are stuck to the outside, the cytosolic face of that endoplasmic reticulum, making it appear rough. Those ribosomes are doing what ribosomes do. They're making proteins, and those proteins the ribosome is making are being fed directly into the endoplasmic reticulum. These proteins can be membrane-bound proteins. They can become embedded in the ER membrane itself, or they can be fed completely through, as this protein has been done, and then that protein would exist fully folded inside the endoplasmic reticulum. We would call the interior of the endoplasmic reticulum the lumen, L-U-M-E-N, the lumen of that structure. The smooth ER makes membranes. It also, or I should say membrane lipids, phospholipids. The smooth ER also makes the steroid hormones that are important for our development. A lot of things that would otherwise be toxic to the cell are sequestered in the smooth ER, and the smooth ER also is the main storage site for intracellular calcium ions. And so this is really what we mean, bringing these 3 structures together conceptually. We have the nuclear envelope made up of its 2 separate layers, its 2 separate membranes. The inner membrane surrounds the nucleus entirely. The outer membrane kind of feeds into, or even more accurately, becomes the rough ER. The rough ER is rough because it's studded with ribosomes on its exterior face, and the rough ER feeds into or becomes the smooth ER. There's really no difference between the smooth and rough ER other than the presence of ribosomes. So that leads us next to the Golgi. The Golgi complex is usually found very close to the nucleus, near the center of the cell. And it too is made up of protein stacks, protein sacs, and lipid stacks, lipid sacs, and lipid tubes. The Golgi accepts proteins and lipids from the endoplasmic reticulum. Usually these are shipped by vesicles, membrane-covered vesicles, and we're going to talk all about vesicle transport in the next lecture after this one. The Golgi modifies those lipids and those proteins, and then it sends them out to their next or final destination in the cell. Lysosomes, talked about those as well. Lysosomes are the stomach of the cell. They're involved in digesting engulfed material. Also, it's important to expand on that a little bit here and tell you that lysosomes also consume and thereby degrade faulty organelles. So if there's been a mitochondrion in the cell that's been there for a long time, it's gotten quite old, it's starting to not do things properly, it's starting to show its age, it's breaking down, that mitochondrion will be engulfed by a lysosome and digested down to its very basic parts, and then those parts will be recycled and used to build new things in the cell. Before anything makes it to the lysosome, the vesicle that contains the material that is going to be digested by the lysosome passes through a series of other membrane-bound organelles that are called endosomes. And endosomes, although fairly well characterized, are one of the most mysterious structures in the cell. The endosome is kind of like an intermediate between a fully formed vesicle that contains something new and a lysosome that is digestive. The endosomes function to basically sort the contents of the vesicles, sending those things that are undigestible back out of the cell and routing things that are useful for the purposes of energy or nutrition to the lysosome for digestion. The textbook takes some time about now to discuss peroxisomes again, but really don't add any new information, so we're going to skip peroxisomes this time. We've covered them sufficiently already. So this table is actually quite a good resource for you in preparing for the next exam, I think. Table 15.1 in your textbook does a very nice job of just listing the different organelles, or they call them compartments here, because that's really what we're doing, is we're compartmentalizing activities. And then gives a quick blurb on the main function for those organelles. We're not going to talk in any depth about mitochondria or chloroplasts in this class because really the whole second half of the typical one-semester undergraduate course in biochemistry is about nothing more than glucose metabolism, and glucose metabolism occurs solely in the mitochondria and the chloroplasts. So when you take biochemistry, if you haven't taken it already, you'll learn all you ever wanted to know about what goes on in these organelles, and then some. And so we won't double up on that information here. Many of the organelles that we find in a typical cell are held in their unique location in the cell by the cytoskeleton, which we've talked about in some depth already. Most of the interactions between the organelles and the cytoskeleton occur between membrane-bound proteins on the organelle and the microtubule components of the cytoskeleton. Organelles make up about 50% of a typical cell's entire volume, and, like anything else, you can tell what's important to the cell by looking at what it gives most of its space to. This is true of any of us. You can tell what we're most interested in based on how much of it we have on our hard drives or how much of it we have on our walls, how much of it we spend our time during the day doing. So, just like us, the cell kind of shows its most important attributes by what it has more of. So we can look at the general average percentage of total cell volume versus what organelle is made up of that volume, and we see that mitochondria is the most important thing to the cell. Having energy, having ATP is a critical need of the cell, obviously. After that, the endoplasmic reticulum. Then the nucleus, surprisingly, then the Golgi, and then the lesser organelles take up about 1% each. So energy is the cell's most important concern, and right after that is the making and sorting of proteins. The total area of membrane found within a single cell is enormous. By this I mean all of the membranes of all of the organelles. The endoplasmic reticular membrane alone in a typical mammalian cell like ours sums up to be 20 to 30 times more than the entire cell membrane itself. This is because it's all folded and convoluted, so there's a lot more surface area there. It's like taking a piece of paper and crumpling it into a very small ball. You can get a lot of paper in a small area. So the endoplasmic reticular membrane is the largest membrane in the cell. In fact, surprisingly, if we look at just the size and the mass of the membranes in the cell membrane itself is one of the minor membranes in the cell. It's actually one of the smallest membranes in the cell in area, in mass. The cell has a hell of a lot more membrane dedicated to mitochondria, endoplasmic reticuli, and of course chloroplasts if it's a plant, than it does to its own cell membrane. So to truly understand the functional relationship between organelles with one another, with each other, we really need to take a step way back in time and begin to understand how these organelles evolved. How they evolved shows us how and why they interact the way that they do now together. And these organelles evolved in stages. The question becomes, which was the first and why? So different membrane-enclosed organelles evolved in different ways, and we're going to leave the story of the chloroplast and mitochondria alone in this lecture. We'll touch on it in just a moment, but we're going to focus more on the organelles we haven't discussed much: the nucleus, the Golgi, and the ER, the endoplasmic reticulum. So the evolutionary precursors of eukaryotic cells are believed to have been very much like bacterial cells. We had said that before. They were probably anaerobic. They were predatory, so they ate other things. But they were just really big, slightly more complex bacterial cells. They didn't have intracellular compartments. They had a plasma membrane, and that was it. In this early pre-eukaryotic cell, the plasma membrane being the only membrane, had to be responsible for all membrane-dependent cellular processes. In fact, that's what bacteria do now. Anything that needs a membrane in a bacterial cell must occur at the cell membrane, because the bacteria doesn't have any other membrane. For bacteria, this works. They're so small, they're so simple, they're so streamlined, that a relatively small membrane is sufficient. But as these early eukaryotic cells grew, or these soon-to-be eukaryotic cells, as they grew, they became more complicated. And being more complicated, they required more membrane-dependent processes to survive. So they needed more membrane to support this complexity. Membranes support life-sustaining cellular functions. We create batteries with membranes. The membrane potential is a stored battery, as we talked about in the last lecture, and that's just one example. So if you're going to be big and you're going to be complicated, you need a lot of membrane. Large cells need more things to do. They need more activities. They need more functions. That means they need more membrane. So it's now believed that these intracellular membranes, which allow these more complicated processes to occur, evolved in two completely independent ways. The two different membrane bilayers of the nucleus, the nuclear envelope, as well as the membrane of the ER, the membrane of the Golgi, the lysosomal membranes, and the endosomal membranes, are all thought to have arisen from the invagination of the cell membrane. That just means the pinching in. So in our early pre-eukaryotic cell, we had the DNA here near the cell membrane. Maybe it was even stuck to the cell membrane, like it is in bacteria, to keep it kind of sequestered in one place of the cell. This DNA had the membrane pinch in around it. See, pinched in, invaginated. Some membrane-bound ribosomes, maybe. These are ribosomes inside the cell that are bound to the membrane. They got pinched in too because they were near the DNA. They were translating close to the DNA, and so they got invaginated as well. And this invagination occurred so much that it actually enclosed the entire DNA molecule of this cell, and it pinched off from the main membrane. So this is just a complete invagination, and then it pinched off. Well, what do we have? We have a number of things. We have a membrane-bound tube that has ribosomes stuck to it. Well, that's an endoplasmic reticulum. We have a nucleus now, but look, because of the way this invagination occurred, this single line is of true bilayer. This invagination actually results in a double membrane around the nucleus. A double membrane because to make this pinching in occur, we needed the bottom membrane and the top membrane here, as shown in this picture. Well, that explains the double membrane of the nucleus, doesn't it? And so in this simple model, we actually explain much of what we see in the modern-day eukaryotic cell. These membranes remain connected to one another because they all arose from the same place. Even more dedicated, more specialized structures like the Golgi and the lysosome and the endosome, they sprang from these initial invaginated membranes. So all these membranes are related to one another. And because of that relatedness, both in function today and evolutionarily, we refer to all of these organelles together as the endomembrane system. And I like that for a number of reasons. One reason I like that is because it groups them all together. Groups all these structures together under a common purpose, and indeed they are related to one another. But more importantly, the endomembrane system suggests that all of these structures rely on their membranes, and that's really the point here. These membrane-bound organelles, these compartments, are nothing more than specialized membranes with stuff in it that allows the membranes to do what they do. It's the membranes and those proteins associated with the membranes that really make these organelles tick. And the organelles ticking the way they should really make the cell survive. With the exception of the nucleus is now a privileged organelle, and it has the nuclear pore, and that's it. With the exception of that nucleus, all of the organelles made up by the endomembrane system communicate with each other and transfer materials between one another through vesicles. Vesicles budding off of one component, one organelle, and fusing with the other. These membranes of the endomembrane system are all compatible to this day. They can trade places with each other from one organelle to the other because they all came from the same place. They all arose from the same evolutionary origin. In many ways, even today, cells treat the inside of these organelles as extracellular because indeed they are. Look at the inside of the endoplasmic reticulum here. Where did that inside come from? Well, it came from the exterior extracellular environment. Whatever is out here in white, that's what's permeating through these membranes. So when these membranes pitched off, they took the outside of the cell with them. And so to this day, the cell still treats the inside of the organelles as outside the cell because that's indeed where they came from. Also supporting this idea is that membranes inside the cell look very much like cell membrane turned inside out. Again, consult with this figure, but it's easy to see why that might be. And finally, further giving this theory credibility, is the idea that even today, modern-day bacteria can be observed to invaginate their membranes, seclude specific related structures, oftentimes it's photosynthetic pigments, photosynthetic molecules, and get those molecules away from other parts of the cell. Essentially, although it's rare, we can witness modern-day bacteria creating proto-organelles by taking some of their cell membrane, invaginating it in, and using that membrane to create a compartmentalized, specialized structure for a specific type of molecule. The other evolutionary origin of organelles, remember I told you there were two different ways that we believe membrane-bound organelles evolved. The other way is the one that we've already discussed. This idea of some prehistoric, pre-eukaryotic cell engulfing but not digesting a smaller aerobic bacteria. That's the other way we got a membrane-bound organelle. Supporting this is that mitochondria, as well as chloroplasts, have their own genomes. We've said that before. They do their own translation and make their own proteins. We've said that before, etc. In fact, this was an exam question on exam 2. Interestingly, all those vesicles that are traded of the endomembrane system, all this trading of membrane and components, this shuttling of vesicles back and forth that goes on in a cell, it doesn't include the mitochondria or the chloroplasts. It's almost as though the mitochondria and chloroplasts in the cell are other. They are not of the same kind, and that's true. If these theories are correct, all of the other organelles are from the endomembrane system. They all arose from the cellular membrane itself. Mitochondria and chloroplasts truly are other life forms, in fact. They existed on their own as independent cells that were then engulfed. So it makes sense that we don't see vesicle traffic go to and from the mitochondria, because that's not where the mitochondria evolved from. Really, really interesting stuff. Okay, so we've kind of opened the door now to this idea of trading proteins and trading components and moving things around, as well as the relatedness between organelles and that they rely on each other's functions. We've set the stage for cells needing a process, needing a mechanism to get proteins from one place of the cell to another, and maybe even more importantly, to sort those proteins, to get the right proteins to the right organelles at the right time. So how do we do that? What gets lost in a lot of the glamour of mitosis and all the steps of mitosis that are covered in genetics is that before you can even get close to mitosis, any eukaryotic cell that wants to divide has to first duplicate and divide its organelles. You see, cells have no mechanism by which to make new organelles from nothing. A cell can't make an endoplasmic reticulum from no endoplasmic reticulum. A cell can't make a brand new lysosome from no lysosome. All organelles must come from pre-existing organelles which grew and then divided in a larger maternal cell. Kind of like the cell theory itself, right? Every single cell has to come from a predefined pre-existing cell. Well, every single organelle in a cell must come from a predefined pre-existing organelle. Also, much like a cell itself, organelle growth requires, well, new lipids to make larger membranes, new protein to allow for a larger organelle to function just as the smaller one did. And so if a cell is going to divide, its organelles need to divide. If the organelles need to divide, the organelles need to get bigger first. If the organelles need to get bigger, they need more membrane, they need more protein. This means getting newly synthesized proteins to the correct organelles is a critical process for something as ultimate as cell growth and division. You need to get the right proteins to the right organelles so those organelles can grow, so those organelles can divide, so that the mother cell can eventually divide, and we can have cell division. Critical component to life. It is this protein sorting and trafficking, making a new protein, but more importantly, getting that new protein to its proper organelle. That's what we're going to spend the rest of the time today discussing. So let's set the stage with some basics. There are 3 different ways by which proteins can enter an organelle. Proteins are delivered directly from the cytoplasm to the nucleus, to the mitochondria, and to the chloroplasts. These are the privileged organelles. These are the organelles that don't participate in vesicle traffic. For the organelles that are part of the endomembrane system, the ER, the Golgi, the lysosome, the endosomes, lipids and proteins are delivered indirectly via vesicles. And these vesicles come from the endoplasmic reticulum. See, proteins enter the ER from the cytosol. But those that are not endoplasmic reticular proteins themselves, in other words, those proteins that aren't meant to spend their entire life in the endoplasmic reticulum, are transported next to the Golgi. And then, if they're not meant to stay in the Golgi for their whole life, they're transported onward to their final cellular destination. This process that I've just described is what we spend almost all of the next lecture discussing. Proteins that are made by free ribosomes that are floating in the cytosol are synthesized containing amino acid sequences called sorting signals, and these sorting signals act as protein address labels. These address labels can either be on the amino terminal beginning end of the molecule of the protein or the carboxy terminal end of the molecule. They're usually on the end, one end or the other. And these sorting signals provide a cellular mechanism to get these proteins to the right place. These signals are specific sequences of amino acids. And these amino acids target the protein to a specific organelle. Once the protein gets to the proper organelle, the sorting signal is usually removed and degraded, and the protein enters that organelle, and that was its destination, so it got to where it was going. There exists signal sequences or sorting signals for all of the major organelles in the cell. There are nuclear signaling sequences, endoplasmic reticular signaling sequences, those for mitochondria, those for chloroplasts, etc. There are proteins that are synthesized that lack sorting signals. These proteins are made lacking a sorting signal on purpose. Any protein that lacks a sorting signal is destined to remain in the cytosol, and there are plenty of proteins that are cytosolic. The proteins of the cytoskeleton are just one example, but many proteins are destined to be in the cytoplasm, and so they're just made without any sorting signal at all. Regardless of the final destination of a particular protein, the single challenge is constant for the cell. If we know where the protein is supposed to go, and we can get the protein there, how the hell do we get a hydrophilic protein across a hydrophobic membrane? How do we get the protein into the organelle? This is what there exists 3 mechanisms for. Nuclear proteins enter the nucleus through its only portal, the nuclear pore. And so proteins that are destined for the nucleus are made in the cytosol and transported to the nucleus through the nuclear pore. We'll expand on all of these mechanisms in just a moment. I said it at the beginning of the lecture, but the nuclear envelope is completely impenetrable. Nothing gets in or out of it. And so if anything is going to enter the nucleus, it has to do it through that pore. Our second mechanism for getting things into organelles, proteins, is that proteins that are moving to the endoplasmic reticulum, the mitochondria or chloroplasts, are transported across these organelle membranes by specific proteins themselves, membrane-bound proteins, called protein transporters. A little bit complicated, but follow me if you can. Protein transporters are membrane-bound proteins which transport other proteins. Okay? So it's like a machine that builds a machine. Okay? It's like a book that describes making books. Protein transporters are proteins that transport other proteins across these organelle membranes. And that's how we get things into the chloroplast, the mitochondria, the peroxisome, and the ER. Unlike nuclear proteins, which enter the nucleus, go through the nuclear pore in their fully folded state, any protein that's going through a protein transporter into the ER, the mitochondria, the chloroplast, has to be unfolded into its primary structure first, has to be unfolded into nothing more than a chain of amino acids, and then refolded once it's made it into the interior of that compartment. So we're going to come back to this schematic in just a little bit and cover the details in a little bit greater depth. But we see already we have our sorting signal, our signal sequence here. This is saying you are destined for the mitochondria. Using that signal sequence, this protein makes it to the mitochondrial membrane, and then it is threaded through as a single chain of amino acids, threaded through a transport protein into the interior of the mitochondria. Once in the mitochondria, that folds up again, folds up into its normal native structure, and now this protein is in the mitochondria where it should have gone. The sorting signal has been cleaved off and degraded, and that protein is ready to do its mitochondrial function. The third mechanism, proteins that leave the ER or are being traded between the different organelles of the endomembrane system are moved by transport vesicles. Transport vesicles are hollow balls of membrane that contain cargo, the cargo in this case being the proteins themselves. These form at the interior face of the membrane, at the origin organelle, the organelle that they started at. They pinch off and then they migrate to their final destination. So we see some proteins here at the endoplasmic reticulum are meant to go beyond, and so we have this pinching off of a vesicle. The vesicle forms, it pinches off, it's carrying our cargo. The vesicle then floats as a ball of membrane to its next destination, in this case the Golgi. And then once at the Golgi, that membrane, vesicle membrane, fuses with the Golgi membrane, and by fusing we release our cargo into the Golgi. And so we have transport through the nuclear pore for the nucleus, we have transport single file unfolded into the chloroplast and the mitochondria, and we have vesicle transport for everything else. Again, when these vesicles fuse with their destination organelle, by fusing those membranes, we pop that vesicle open, we pop it open on the interior side of the destination organelle, and so the contents of the vesicle are spilled into it. Alright, so back to signal sequences now, these sorting signals. Let's talk a little bit more about them. A typical sorting signal, and again, as I've already been doing, they're more commonly referred to as signal sequences. A typical one is between 15 and 60 amino acids long. These signals are very potent. They are necessary and sufficient for correct protein trafficking. What we mean is that if a protein contains the right signal sequence, it goes to the right place, period. It needs nothing more to get there. And without a proper signaling sequence, you can't get the protein to the right destination. So you it is necessary to have the right signal sequence to get where you're going, and it's sufficient. That's all you need to get there. So in other words, to be correctly trafficked to your right destination, a protein needs the correct signal sequence, and that's all it needs. We scientists, the field, biology, have figured out most of these signal sequences, and your textbook gives an example in Table 3. You can see that there's import into the endoplasmic reticulum, other proteins that are meant to stay in the endoplasmic reticulum for their whole existence, import into the mitochondria, import into the nucleus, import into peroxisomes. All of these have these well-defined signal sequences. Pop this sequence onto the end, in this case the amino terminal end, of any protein you want, and that protein is going to head directly into the ER. This is easily shown experimentally, and in fact I'm going to ask you in class the next time we meet to tell me how you think that works. It appears to be the chemical property of the amino acids in the signal sequence that plays the most important role, as opposed to the amino acid sequence itself. In other words, if there is a charged amino acid somewhere on here, like this lysine is positively charged. If you were to take this lysine out and replace it with another positively charged amino acid, the signal sequence works just fine. It's not important that lysine is here, it's important that a positive charge is here, in other words. So it's the charge, it's the hydrophobicity, it's all of the chemical properties of the sorting signal that tell the cellular machinery where to bring this particular protein. Not the amino acid itself, but its chemical properties that do that. So, for nuclear proteins actually have to get through two different membranes. The two membranes of the nucleus are actually quite different. The inner membrane is often connected to a protein meshwork on the interior face of the nucleus that's almost like a nuclear cortex, similar to the cell cortex we've already described, but this time inside the nucleus. Chromosomes themselves are also often attached or connected to the inner face of the nucleus. The outer membrane of the nucleus, as we've already said, springs from or becomes the endoplasmic reticulum. So those two membranes are very, very similar. You can see in this diagram from your textbook, this green mess here is meant to indicate that nuclear cortex, that nuclear framework or meshwork of proteins inside the nucleus. And you can see the nuclear membrane is in contact with that meshwork. The outer membrane of the nucleus, of the nuclear pore, feeds directly into the endoplasmic reticulum. And then we have these nuclear pores, these hollow entry points into the nucleus. So the nuclear pores are the gates into the nucleus, and they're the gates through which all molecules must pass if they are going to enter or leave the nucleus, because the nuclear membrane is completely impermeable. Newly made nuclear proteins can enter from the cytosol where they were made. Newly made messenger RNAs can exit the nucleus to the cytosol from where they were made. And remember, this export of mRNAs is actually a control point for messenger RNA fidelity. We talked about this in our Central Dogma series of lectures. The ability for a messenger RNA to leave the nucleus is how we check that it has been correctly tailed, correctly capped, and correctly spliced. Nuclear pores are no joke when it comes to complexity. These are amazing structures. They are elaborate, they are huge, they're filled with water, so nothing can pass through a nuclear pore unless it's hydrophilic. And this is a pretty good representation of what a nuclear pore looks like from your textbook. These structures are so big that you can actually image them with electron microscopy. So this is a true picture from an electron microscope, an electron micrograph of nuclear pores. We're looking down into the top of the nuclear pore. So we are, technically, we're in the cytoplasm looking at the outer surface of the nuclear membrane and seeing these nuclear pores studded. Whereas in this view, we're looking sideways. Small hydrophilic molecules can pass right through the pore non-selectively. They can float right on through these holes. Things like water, like oxygen, like carbon dioxide. They can diffuse into the nucleus, but they have to go through the pore to do it. And they can go in either direction. They follow their concentration gradient. Larger molecules, on the other hand, get caught up and entangled in this web of disordered protein, this web of disordered amino acid chains that fills the interior of the nuclear pore. And this web of proteins, this web of amino acids, is made up by the pore proteins themselves, the proteins of the nuclear pore. Larger molecules that want to get through the nuclear pore and do not want to get caught and entangled in this web must have a signal sequence. In this case, we call it a nuclear localization sequence for obvious reasons. If a protein is in the cytoplasm and it has a nuclear localization sequence in it, other cytoplasmic proteins called nuclear transport receptors will bind to them. Actually, they'll bind specifically to the nuclear localization sequence. These receptor proteins then act as guides. They interact with these fibrils of the pore, with these little finger-like projections of the pore. By interacting with those fibrils, they move this net out of the way, this meshwork, this entrapment, this web that would have otherwise entrapped a protein. Moves them out of the way. And the nuclear transport receptors carry their cargo, the protein we're trying to get into the nucleus, fully folded right through that pore. So I think this diagram does a pretty good job of that. Here's the nuclear localization signal. It's an amino acid sequence on the very end of this protein. This protein wants to get into the nucleus where it belongs. This nuclear localization signal is a point of attachment or point of recruitment for a nuclear transport receptor. So the receptor binds directly to our protein through that localization signal. The receptor protein then interacts with the fibrils, pulls on them and pushes them, and by doing so moves this protein net out of the way. Then together the receptor as well as the protein we're trying to get into the nucleus float into the nucleus. Literally, our protein is escorted into the nucleus by the receptor. Once in the nucleus, the two proteins disengage. Our nuclear protein is now nuclear, hooray! And the nuclear receptor, the transport receptor, turns right around and recycles right back out through the nuclear pore, back into the cytoplasm, so it can repeat the process and escort the next nuclear protein in. So once everybody's inside, the receptors let go and recycle back out. Beautiful little system, no? Oh, really nice way to get proteins specifically into the nucleus only when they belong there, through this horribly complicated protein structure of the nuclear pore. Okay, so on now to the mitochondria and the chloroplasts. Very similar mechanisms for both of these organelles, and as we've already mentioned, proteins need to unfold into their straight amino acid chain, their linear chain, in order to enter these organelles. We've said in other lectures that the structure of the mitochondria and the chloroplasts are quite complicated. Mitochondria has two separate membranes that make it up, an inner membrane and an outer membrane. Chloroplasts actually have three membranes. And you'll see why in other classes why so many membranes are necessary, but it creates a bit of a challenge for transport. You not only have to get through one membrane, you have to get through two or more. Also, while it is true that the mitochondria and the chloroplasts have their own genomes and code their own genes and synthesize their own proteins, it's important to note that over the course of evolution, a lot of the mitochondrial and chloroplast genes have, believe it or not, left these organelles and migrated into the nucleus. So we find genes in the nuclear DNA that had previously been part of the mitochondrial or chloroplast genomes. So mitochondria and chloroplasts can't make all of the proteins they need. Some of the proteins that these organelles need are made by nuclear genes, by cellular translation, and then those proteins need to make it back into their proper organelle. So these proteins that are required for these organelles to function, but are made by nuclear genes in the cytoplasm, must be transported into these organelles from the cytosol. There are specific locations in each of these organelles, mitochondria and chloroplasts, where the two membranes of the organelle touch, they meet. And it is at these junction points of these membranes where the proteins are threaded through. So this is the figure we looked at before, and now we're going to go into it in a little bit more detail. But you can see this is the outer membrane of the mitochondria, and here is the inner membrane. And at certain junction points, contact sites, these two membranes touch. And even more importantly, channels or transport proteins for these two membranes come in contact. So this is an entry point into this organelle now. This is a way to get from the cytoplasm into the interior of the organelle. We have a signal sequence on our protein of interest. It's a sorting signal. It's specific for the mitochondria in this place. It's amino-terminal, so it's at the very beginning of the amino acid chain of the protein. And this signal sequence is recognized by a receptor protein found on the outer membrane of the organelle. So here's our receptor protein in blue, and you can see there's a direct protein-protein interaction between our sorting signal in red on our protein of interest. That's the sorting signal that says, hey, I'm mitochondrial. Bring me to the mitochondria, that's where I go. This sorting signal interacts directly with a receptor that's only found on the outer surface of the mitochondria. This is a direct linkage here, bringing our protein of interest to the right membrane. Then, once that interaction is made between the receptor protein and the sorting signal, the protein begins to be threaded through the protein transporter. The protein transporter here is in yellow. This is a membrane-bound protein that moves or transports other proteins. And here is our protein being threaded through these transport proteins into the interior of the mitochondria. Because it's being transported as a single chain of amino acids, the protein has to unfold in order to undergo this transport. Once that unfolded protein makes it completely inside the organelle that it is destined for, the signal sequence is removed. It's no longer needed. That signal sequence was just an address label. That signal sequence was just an amino acid chain that said, bring me to the mitochondria. Once you've gotten to the mitochondria, you don't need the label anymore, so you chop it off and degrade it, reuse those amino acids for something else. But we're still stuck with this unfolded protein. Other additional proteins called chaperone proteins do what chaperones do. They accompany our protein, make sure our protein isn't doing anything naughty, isn't doing anything that it shouldn't be doing, and helps our protein fold back into its correct three-dimensional shape. It's important to note here, although we'll cover it all a little bit more in the next lecture, although very, very briefly, that new membrane phospholipids are brought to the organelles that we're discussing here, to mitochondria and to chloroplasts, from the endoplasmic reticulum, not through vesicles. Remember, vesicles don't communicate with these non-endomembrane components. Vesicles don't trade with the mitochondria or the chloroplasts. So new phospholipids are brought to these organelles to make new membrane through specialized carrying proteins that carry these phospholipids singly, one at a time. So that's how we get proteins into the mitochondria or the chloroplasts. How do we get proteins into the endoplasmic reticulum? Well, that's an even more interesting story. Unlike everything we've discussed so far, the endoplasmic reticulum serves not as the destination of a protein, but most often as its origin, as its birthplace, as its start. And then from the endoplasmic reticulum, these proteins go to their other places. They usually head first from the ER to the Golgi, and then they can go on further to the endosome, the lysosomes, the cell surface. They can be completely secreted. Of course, some of these proteins are endoplasmic reticulum proteins, so they stay in the ER. Others are Golgi proteins, and they stay in the Golgi. But all of these proteins have quite a journey ahead of them after they've been made in the ER. So just to summarize that idea, we're about to make proteins at the ER, into the ER, and from the ER they will either remain, they'll go into the Golgi where they'll stay, or they'll go to the Golgi and from the Golgi go to secretory vesicles where they'll be released from the cell entirely, lysosome and endosome maybe, or they are membrane-bound proteins and they will migrate from the Golgi to the cellular membrane. It's important to note, almost sadly, that once these proteins are in the ER, they are treated as extracellular by the cell. They are other. So once a protein is in the ER, that protein will never again see the cytoplasm. It will never be cytosolic again. Regardless of where it goes next, it will be in an enclosed compartment or released from the cell. The way that those proteins will avoid the cytoplasm from this point on is that these newly made proteins will be ferried from destination to destination, from organelle to organelle, in transport vesicles, again, to be talked about in depth in our next lecture. Hydrophilic, free-roaming proteins enter the endoplasmic reticulum completely. These are non-membrane-bound proteins, proteins that contain no hydrophobic regions in them at all. They exist, folded up again, in the interior portion of the endoplasmic reticulum. That, again, is the lumen of the ER. Transmembrane proteins are made in a very similar way. They are also fed into the ER, but while being made, these proteins become bound to the endoplasmic reticular membrane. This is accomplished by a partial translocation through that membrane. Once in the ER membrane, those membrane-bound proteins can be brought to other organelles where they're membrane-bound there, or be brought to the cell membrane and be cellular membrane-bound proteins. But once a membrane-bound protein is embedded in the ER membrane, it will never be out of a membrane again. All of these proteins, whether they're free and hydrophilic or membrane-bound, are initially directed to the endoplasmic reticulum by, of course, an endoplasmic reticular signal sequence, an ER signal sequence, that's found at, again, the amino terminus of the newly made protein and directs that protein to the ER. However, unlike any of the other proteins we've discussed thus far, proteins that enter the ER begin doing so before their translation is complete. So this is a coupled process. The way that it works is that the translation of a protein, of any protein, begins at a free ribosome floating around in the cytosol. However, if the new protein being made by that ribosome contains an ER signal sequence, remember where the ER signal sequence is. It's at the amino terminal end, the very beginning. So as the very beginning of that amino acid chain emerges out of the back of the ribosome, if that chain is an ER signaling sequence, that amino acid chain is brought directly to the cytosolic face of the ER. And the ribosome, which is still chugging away at translation, is brought along for the ride. So now we have the amino terminal end of our protein bound to the ER membrane, and with it comes the ribosomes. This is the origin of the rough ER. The rough ER appears rough because ribosomes are associated with the cytosolic face of the ER membrane. But those ribosomes are there only because these endoplasmic reticular proteins are being recruited to the membranes themselves. This is what makes the rough ER rough. There are no ribosomes that exist solely at the endoplasmic reticular membrane. It's not as though there are two classes of ribosomes, some that are free in the cytosol and some that are at the membrane. All ribosomes are free ribosomes. It's just that many of them are brought to the endoplasmic reticular membrane because the proteins they're making happen to be targeted for the endoplasmic reticulum. It's really, really interesting stuff. Okay, so once that protein is done, that ribosome doesn't have anything holding it to the membrane again, and it floats away back into the cytosol. It returns to being free. Again, there are no such thing as endoplasmic reticular ribosomes. But that's really not what we're after here. What we're trying to talk about is how proteins themselves get transported into the ER. So let's discuss that process in some more detail. The ER signaling sequence, again found at the amino terminal end of our newly made protein, is bound by and recognized by something called a signal recognition particle. This is a protein as well. The signal recognition particle is very much like the nuclear transport protein that we talked about when we were discussing the transport across the nuclear pore just a moment ago. This is a protein, the SRP, which binds to the ER signal sequence. So here's the SRP in brown. And you can see our ribosome is on our messenger RNA here, chugging away, making new protein. First thing to come out of the back of the ribosome is the ER signaling sequence. And as soon as that ER signaling sequence emerges out of the ribosome, it is bound, pop, by the SRP. The SRP binds directly to that sequence. The SRP can interact with another thing as well and simultaneously. While interacting with the ER signaling sequence on one end, the SRP is also interacting with an SRP receptor found on the surface of the ER membrane. Here is our SRP receptor in blue, interacting with the other end of the SRP. See what happened here? The SRP is a bridge. The SRP is bridging the newly made protein with the endoplasmic reticular membrane. That's what the SRP is doing. The binding between the SRP and the signaling sequence on the newly made protein slows the ribosome down, allows the SRP to find and interact with its receptor, and more importantly, allows the SRP to be released and the newly synthesized amino acid chain to be threaded through a protein translocation channel that is on the surface of the ER. This is a membrane channel. This is a hollow opening into the interior of the endoplasmic reticulum, and once the SRP binds to the receptor, all of those components are released, and the newly made amino acid chain is threaded directly through that channel. into the interior of the ER. So we needed the signal sequence on the newly made protein here in red to attract the SRP in brown. We needed the SRP in brown to bring us to the receptor in blue. That created our rough ER. That recruited the ribosome. We needed that interaction between the SRP and the receptor to thread our newly made protein into the translocation channel. But once that threading is done, we don't need the SRP anymore, and we don't need the receptor. Now, as this protein continues to be made by the ribosome, it will be fed through this channel into the interior portion or the lumen of the ER. This is a perfect example of a step-by-step, protein-by-protein-based biological process. The ER signaling sequence, once recruited to the translocation channel, keeps that channel open, allowing the rest of the protein to be threaded through. Interestingly, since the ER signaling sequence is keeping the channel open, that means it remains bound to the channel while translation is taking place. Translocation of the protein occurs with the ER signal sequence bound to the channel. So this is a very accurate diagram. The signal sequence again is in red. You can see that it is prying the channel open, keeping that channel open, allowing the green rest of the protein to be threaded through. But to keep that doorway open, you need to stand there with your foot in it. You need to pry it open. And so the ER signal sequence stays associated with the channel while translocation is going on and translation is occurring. This means that the newly made protein is being translocated in a looping fashion because we have one end fixed to the channel while we thread in the rest of the protein, making the next end. During this process, once the protein is completely translocated into the ER, the signaling sequence is chopped off because we don't need it to hold the channel open anymore. It is then popped out of the channel because the channel closes with the signal sequence shortened. And that signal sequence will eventually be degraded and recycled. And of course, the protein now, not attached to the signal sequence and no longer attached to the channel, is fully in the ER lumen and can fold back up into its shape. So that's how our hydrophilic proteins get into the ER. Our next question is, how do transmembrane or membrane-bound proteins get into the ER membrane? For proteins that are destined to become membrane-bound, and this can be membrane-bound at the ER as well as any of the organelles or even cell membrane-bound. Remember, all of these proteins begin their journey at the ER and then they're shipped to their correct location by vesicles. So in order to make a membrane-bound protein, it needs to first become bound to the ER membrane. That process is very similar to the one we just described, with just a few differences. The simplest example, and the one that we'll start with first, is a transmembrane protein where only one portion of the protein is actually embedded in a membrane. These proteins still have an endoplasmic reticular signal sequence at their amino terminal end. Translocation begins the same way as before. SRP binds to that signal, recruits it over to the receptor. We begin threading this protein into the ER membrane. Translocation occurs. However, somewhere in the middle of that process, somewhere in the middle of that protein, is what we call a stop transfer sequence. That overcomplicates it. The stop transfer sequence is nothing more than a part of the amino acid chain made up primarily of hydrophobic amino acids. That's it. So the stop transfer sequence is just a hydrophobic region of the newly made protein. That's it. However, once the stop transfer sequence makes it into the translocation channel, it stops translocation. Translocation halts. Everything is kicked out of the channel. The stop transfer sequence becomes embedded in the hydrophobic region of the lipid bilayer of the ER. So once this hydrophobic region of this protein, once this stop transfer sequence enters the channel, it's kicked out of the channel, and it becomes embedded in the interior of the ER membrane, which is also hydrophobic. And so this portion of the protein gets stuck in the membrane. The hydrophobic region, I've already said, is released by the translocation channel. It's now free to laterally diffuse throughout the entire ER membrane. Translation continues. The ribosome continues to make the rest of the protein, but now that protein's not being threaded into the lumen of the ER. We're making the rest of the protein on the exterior surface of the membrane. And so once this becomes embedded here, the rest of the protein needs to be made this way feeding off of the cytosolic face of the ER membrane. The signal sequence is released, it's cleaved off and degraded as before, and we've got ourselves a transmembrane protein, right? This is a transmembrane protein. Embedded into the membrane of the ER, we've got a cytosolic side of the protein, and we've got an in this case, an intra-ER side, a lumen side of the protein. But if we ship this membrane-bound protein to other organelles to the cell membrane, it's going to be bound to the membrane there, free to laterally diffuse. Sometimes the ER signal itself is internal, and then we call it a start transfer sequence. It's not at the amino terminal end of the protein, but instead in the middle of the amino acid chain. When this is the case, we still start the process in a very similar way, but we actually haven't threaded the protein into the ER lumen from its beginning. So the amino terminal end of the protein actually hangs out here. In addition, the start transfer sequence is never removed or degraded. It remains part of the protein for the life of the protein. This strategy of translocation is typically used for proteins where multiple regions are embedded in the membrane. I believe Jen Davey asked in our last class about not in our last class, the class before, about the alpha factor receptor, and I mentioned that it was a 7-transmembrane repeat protein. This is how we make such proteins. This is how we thread proteins into a membrane. This is a 2-transmembrane repeat protein here because we have the start transfer sequence embedded in the membrane. This is a hydrophobic region that stays stuck in the membrane. And the stop transfer sequence is also embedded in the membrane. But we can have another start transfer sequence elsewhere in the protein, which would create a 3rd region embedded. And then we would loop everything from that point on into the lumen side of the ER. And then if there was another stop transfer sequence, we would stop threading at that point and keep exterior for the rest of it. That would be a 4-transmembrane sequence. We could have a 3rd start transfer sequence, and we thread it again. And then we loop everything inside, loop, 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 loop. And then there would be a 3rd stop transfer sequence. Makes us stop that looping and put the rest of the protein from that point on back on the exterior side of the ER, and that would be a 6-transmembrane protein. So proteins such as this have multiple start and stop transfer sequences, and that combination of start and stop transfer sequences literally allows these proteins to be threaded or stitched through the membrane, just like stitching up a pant leg. So a start transfer sequence gets you going, and a stop transfer sequence gets you out again. And start transfer sequence gets you going in the first direction, and the stop transfer sequence gets you going on the opposite direction. Start gets you sewing in, stop gets you sewing out. Start gets you sewing in, stop gets you sewing out. And so it's a perfect way to weave protein domains in and out of a membrane. Really elegant system. Whether you're a single transmembrane protein, 2-transmembrane protein, or a 7-transmembrane protein repeat, once you're embedded in the ER membrane, that protein that's embedded must be sent to the organelle or the cell membrane that it's meant for. This involves shipping. In fact, even free proteins that were completely hydrophilic and made it into the ER lumen must be sent to their final destination. This shipping and transport requires vesicles. And vesicle transport is what we will cover in depth in our next lecture. So let's summarize what we've talked about today. We started very basically talking about the different organelles, the membranes around them, and the idea of trying to get proteins in and out of them. We started with the idea of the endomembrane system. The outer membrane of the nuclear envelope is actually continuous with the endoplasmic reticulum. Intracellular membranes probably evolved in 2 independent ways. The membranes of the nucleus, the ER, the Golgi, the lysosomes, the endosomes, all of them likely arose through an invagination event of the cellular membrane itself. And this is why we see a continuity and a relatedness between the nuclear envelope and the ER, among other things. These membranes all now form what we refer to as the endomembrane system. The other way that intracellular membranes evolved was through this engulfment. Mitochondria and chloroplasts are separate organisms that were swallowed, engulfed, but not digested, and so they came with their own membranes and they kept their own membranes. Proteins, once they're made, have to get to their particular destination. Regardless of what that final destination is for a particular protein, the single challenge is how do we get that protein across the membrane of the organelle it's meant for. Proteins are sometimes synthesized containing sorting signals. These act as address labels. These sorting signals are also referred to as signal sequences, and they target particular proteins to particular organelles. Any protein that's made without such a signal is destined to be free and cytosolic for its entire existence. In this lecture, we discussed two of the three mechanisms of transport for proteins. Nuclear proteins have to go through the nuclear pore to pass the nuclear envelope. We talked about that in some detail. And proteins that are going to go into the endoplasmic reticulum, into the mitochondria, or into chloroplasts have to be threaded. They are threaded in an unfolded way through protein transporters. Unlike nuclear proteins, which can remain folded in their three-dimensional structure, any protein that's going to be transported into the ER, the mitochondria, or the chloroplast, has to be unfolded first. We talked about each of these processes in some detail, spending the most time on the endoplasmic reticulum, of course. And finally, it's important to note that all proteins, regardless of where they're going, if they are part of the endomembrane system, they begin their journey at the ER. Truly hydrophilic, free-existing proteins are made and threaded completely into the lumen of the ER, and then they will ride vesicles, transport vesicles to their final destinations. Membrane-bound proteins, and I don't care if they're ER membrane-bound, organelle membrane-bound, or cell membrane-bound, they too begin their journey at the ER. At the ER, they become embedded in the ER membrane first, using stop transfer sequences, and then once those proteins become part of vesicles, they are transported to their final destination. So it's almost like we leave this entire lecture as a ... to be continued, because the very next thing we need to talk about is the third mechanism of protein transport, that is the transport of vesicles, and that will be the topic for our entire next lecture.